All right, we're going to go ahead and get started now. We just heard excellent work from the bench and from mouse models, and now we're going to be moving on to work in highly translational models of HIV infection in the non-human primate realm. Our first speaker is going to be Janice Clements. Dr. Clements is professor of molecular and comparative pathobiology at Johns Hopkins University School of Medicine. She also holds the title of Mary Wallace Stanton Professor for Faculty Affairs and has served as the Vice Dean of Faculty since 2000. Dr. Clements has been involved in HIV research since its inception and was the first scientist to report that HIV is a lentivirus in the early 1980s. Dr. Clements' research focuses on the molecular virology and pathogenesis of lentiviral infections with an emphasis of animal models of viral latency and study of the latent reservoirs in brain macrophages. Today, she will speak on functional latency in CD4 lymphocytes and macrophages, and macaques latently infected with SIV. It is my privilege to welcome Dr. Clements.
Well, I want to thank the organizers for inviting me. It's always fun to speak at home and to all the friendly faces that I know. So while I'm the first name on this presentation, I couldn't have done this work without the entire retrovirus lab. You don't do non-human primate work by yourself. You really need a village, a town, and you need a wonderful supporting institution. We actually have incredible support for our animals here with primate colonies here. Probably many of you don't know that, but we have both a rhesus macaque colony that goes back to the 50s and a pigtail macaque colony that the retrovirus lab built. So we have incredible resources, and I have to, right from the beginning, thank the institution for allowing us to be able to do this. So I think I want to focus on what the current issues for HIV cure research are. Many of us just came back from the Cure Symposia that was just last week. And we all know that the CD4 T cell reservoir is really the major focus. But I think the question is it the only target for eradication? We know that monocytes and macrophages get infected during acute infection, cause tissue-specific diseases like CNS disease, lung disease, cardiac disease. So we know that they get infected, but are they a functional latent reservoir like the T cell? Do they harbor virus and remain latent during ART suppression? And should they be included in the reduction or elimination of latent reservoirs as a strategy for a cure? So just to remind us that back in the 80s, HIV infection of the brain was really a big concern, and it was really a big concern for all the tissue-specific infection of macrophages and tissues until we had antiretrovirus therapy. And so the brain was massively infected, and there were macrophages in many tissues, lung, spleen, cardiac disease is caused by infection of macrophages. So in the last 5 to 6 years, we've learned much more about macrophages too. So a macrophage isn't necessarily a macrophage, it's what the source of that macrophage is. So in tissues such as the brain, the alveoli macrophages, spleen, some of the spleen macrophages, as well as some of the intestinal ones, are derived from embryonal cells. And that means that they can self-renew. So they're different than the monocyte-derived macrophages, which circulate through the body, populate tissues, but they are a different type of macrophage. They're not self-renewing like the tissue macrophages. So in the brain, the microglial cells, which are the embryonal macrophages, it's been shown in mouse models you can completely wipe out those macrophages, microglia, with targeting the CSF1 receptor, and they will repopulate within 2 weeks. So those cells can actually repopulate. So if they're there long term, that could constitute a long-term latent reservoir if those cells are infected. So just some background, we've looked in both, in our models in spleen, and we showed longitudinally that in spleen infected long-term, that macrophages are infected in both the red pulp, which are the embryonal macrophages, as well as the germinal centers, and that those red pulp macrophages don't die even after long-term disease. So there's only a very low level of apoptosis, caspase 3 cleavage in macrophages in the spleen. We've also showed the same thing on the bottom in the brain of SIV macaques on ART. While you can see staining on the left side for caspase 3, very little cell death in the brain. And on the right side, you can see CD8 marking the microglia, and there's even the low level of caspase 3 is not associated with the microglia. So our tissue macrophage is a functional latent reservoir. I think I went over most of this, and what I'd like to show you is how we've developed not only an assay for the functional latent macrophage, but as a prequel to that, we've developed over the last 6 years a T-cell QVOA that measures the T-cell reservoir. So I'm going to talk about actually both models that we're using right now. For the last 10 years or so, we've used the stool inoculated model, in which we put 2 viruses into pigtail macaques, and we get rapid disease, rapid CNS disease, but even that rapid and high level of virus replication is completely suppressed with antiretroviral drugs. Currently, we're using the injectable drug regimen that is a 3-drug regimen, and it completely suppresses virus replication. And so in both this is a representation of the dual infected model, I'll also show you the SIV MAC 251 model, in which we look at both plasma now and CSF, and you see the same thing. So based on the assay that the Silicanos developed, measuring the functional latent CD4 reservoir, we developed, in collaboration with Bob and Kai Dang and Jason Donoso in his lab, a CD4 functional latent reservoir. And what we showed that in the plasma of does this pointer work? Can you see it? I can't see it from up here. Oh yeah, I see it now. It's light. So on the left-hand panel on your side, you see blood, and you see the CD4 lymphocytes, the QVOA for the CD4 lymphocytes. And as you see in humans, you see that it starts off after suppression at a high level, and as you measure that latent reservoir, it decreases and then it plateaus. So we've done this long-term in monkeys, longer than this, and we can tell you that the latent reservoir of CD4 cells in both macaque models is the same as in people, 1 cell in 10 to the 6th. And if you look in tissues on the right-hand side, you see the same thing, head lymph nodes, gut lymph nodes, and spleen, and PBMCs. They all are latently infected. The CD4s are latently infected and at about the same level as in people, 1 in 10 to the 6 cells. As I said, we've spread our wings and developed the SIV MAC 251 model in our lab. We got the virus from Ron DeRozier and made a virus that genetically looks just like other viruses that are used in other labs. And what we find is that, like in other reports, SIV 251, you see it in the plasma, but unlike most studies, we've also looked at the CSF. I think this, again, is one of the reasons to be at a place where you have scientists who can do it, and so our veterinarians have perfected getting CSF from our animals. And so we can get CSF, whenever we get plasma, we get CSF, and so we can follow it. And I think this is the first time that SIV-251 has been looked at longitudinally in the CSF without CD8 suppression or CD4 suppression. And as you can see, all the animals have virus in the CSF. So it's a model for both infection in the periphery and in the CSF. And as I said, under that, we've suppressed with the three the three regimen drugs, and we get rapid suppression. And when you model suppression in plasma and CSF, it is much more consistent in the plasma than the CSF, because the plasma is undoubtedly seeding the CSF, and so you can see it shows more variability, and it's consistent with the animals that we see higher levels if you look really closely at the seeding of the virus. When you look at latency in resting CD4 cells, in the spleen and the PBMCs of the SIV MAC251, you see the same thing as I showed you in our other model. It's the same as in humans, 1 cell in 10 to the 6th. So we've reproduced the T cell assay, so now we know that CD4 cells, resting CD4 cells, are at the same level as in humans. So now the question is there a latent reservoir that is functional? That is, can you get virus out of monocytes and macrophages, out of suppressed monkeys? So we developed this quantitative viral outgrowth assay. Now for macrophages, this is much more complicated. First of all, you have to get the macrophages out of tissues. So for tissues, we have to use enzymatic digestion. And for each tissue, we have a somewhat different protocol, because each tissue has a different array of fiber. And so for each of the tissues, brain, spleen, lung, and other tissues, we use digestion. And then we select those cells by CD11B selection. And we find, at least in the monkeys, CD11B is the best marker to pull out the maximum number of macrophages from all of the tissues. So from brain, spleen, all of the tissues, we pull out macrophages. Now, one difference between macrophages and T cells exponentially grow. When you put 1 million T cells into a QVOA, they exponentially grow during the assay period. When you put 1 million macrophages or monocytes into the QVOA, you get only those number of cells. So there's no exponential expansion of the cell that contains the virus. And like in the CD4 QVOA, we've added CEM-X7D4 cells. Cells that we know are responsive to SIV macrophage-topic viruses, to expand that virus. But it still doesn't make up for the fact that the cell doesn't really proliferate. To make sure that all we have in the assay are macrophages, because I think one of the arguments could be is that you're taking macrophages out of tissues that also have T cells. So we've been very rigorous, and we developed a TCR, a beta RNA probe, and for every sample, we plate the macrophages, the same number of macrophages in the second dilution of the well, and we do TCR beta RNA analysis on those cells. And we never detect enough T cells in those wells to account for the data that I'll show you for latency. Let's see. Let me go back. So we allow those cells to, in the case of macrophages, to adhere, and then we add the CEM-X7D4 cells. For monocytes, you actually have to let the monocytes mature for 7 days, and then you add the cells. This just shows you, because that was a major question we were asked, do the macrophages stay alive in culture? Do they die off? Do the CMX74 cells overgrow them and kill them? And so what we show you here is that by using Frodo-labeled E. coli, that the macrophages, after 14 days, are maintained in culture, and they're alive. So they're functional, they phagocytize the E. coli, and they are alive and stay alive for the 14 to actually 21 days if we go that long. So they stay alive. So this slide shows the comparison of the SIV-251 animals, in which on the top we show the tissues, RNA and DNA from the viremic animals, and then from the RNA and DNA from the suppressed animals. And why I'm showing you this, because it's very important that if we're saying that there's latency in macrophages, that we actually have no RNA in the tissues. And what we've shown is that the RNA is substantially decreased to very low levels, but the DNA levels in the tissues are relatively unchanged. And then when you look at the QVOA, so the macrophage QVOA in those animals, and we can detect the microglia and perivascular macrophages in brain, in spleen, in lung, and in PBMCs in blood, and they're all approximately at 1 in 10 to the 6 cells. In this case, these were, the brain macrophages were a little bit higher, but it's not really significantly higher when you see the spread of this. So it's at the same level as T cells, 1 cell in 10 to the 6th, and I have no explanation for that. We were really surprised at that. I mean, we really guessed that it might be lower and much harder to detect, but in fact, there's the same level of latency in the macrophages in both models. When we've looked at both the dual inoculated model, which is a more macrophage tropic virus, we have 1 virus in there, there's no difference between the 251 model and this model, the 251 and the dual inoculated. When you look at CD4 cells in the blood, what you see is that the monocytes, I'm sorry, this is the spleen. I'm looking up. In the spleen, you see that the CD4 cells and the macrophages in the spleen, again, have a very similar level of latency in the spleen and also in the PBMCs. So the monocytes and macrophages harbor latent a reactable virus at the same frequency, 1 in a million cells, at least in the monkey model. So our conclusions is that macrophage QVOA identified a functional latent reservoir and suppressed macaques in monocytes in blood and macrophages in spleen, lung, and brain. The frequency, and this is, I find surprising, and probably it will tell us a lot about latency of HIV and SIV in cells. The frequency between resting CD4 cells and macrophages is the same, and the macrophage QVOA is applicable to analysis of human monocytes as well. So we've adapted it so that we can look at that. So I just want to show you a little bit more data on the viruses that we're looking at. Again, this is a dual inoculated monkey that was injected with ART and with Maraviroc. Unfortunately, Maraviroc is a CCR5 blocker. We expected to see potentially a better response and a better reduction of virus, but really it looks very similar. And when we did QVOA with the Maraviroc versus the injectable only, again, what you see is that the macrophages, which you would expect to be prevented from being infected because of the CFAR blocker, there was the same level of latency in macrophages with and without Maraviroc. When you allow these animals to rebound, what you see is that the animals rebound at different rates. And what we found was, and they rebound both in the plasma and the CSF. And if you notice, the animal that rebounded highest and more rapidly in the plasma was also the animal that rebounded in the CSF. And when you analyze the data, what you see is that the slower the suppression of the animals, the longer it takes. So on the left side is the time to suppression. And on the right is the time to reactivation. So you can see that, at least in plasma, it really carries through that the longer it takes to suppress the viral load, and probably the larger the reservoir, the more rapidly there is a rebound of the virus. And it's also true to some extent in the CSF, but not as strictly as we see in the plasma. But the animals that suppressed faster came up later than those that, and we're pursuing these studies with the QVOA as well. We've done the same thing, rebound, in this SIV MAC251 animals. And again, what you see is that there are animals that rebound more rapidly. And in the case of the SIV MAC251, we found only one rebounded in the CSF, and that was the animal that rebounded rapidly and to the highest level. So I'm showing this to you because I think this gave us some insight into what happens after rebound. So I think what we all expect is that after rebound, you would see increased numbers of both CD4 cells and macrophages, if they are a latent reservoir, in these tissues. And so we did longitudinal QVOAs at the first bleed that was suppressed, a later bleed, and then at necropsy. And you can see, as what you might expect, in the animals that rebounded, all of them rebounded, you see that the frequency of latently infected cells go up. Then when you look at the tissues, the same thing. We actually do spleen biopsies mid-level of suppression, and then we do it at necropsy. So the biopsy was low, what you expect, about one in a million cells of T cells, but there was far more T cell activation and latently infected cells in the rebounded animals. Surprisingly, we didn't see the same thing from the monocyte macrophages. And again, I think this is telling us something about what's going on in these cells. So when we look at the blood cells, what we saw was a very, the subsequent bleeds of QVOAs, we see that the latency in monocytes is relatively stable. It's really surprising, and I think this is something that we're going to have to really look carefully at. And in the spleen, where we saw it went up very high for T cells, the macrophages in the spleen, in fact, did not go up. So this is surprising, but it also may be that just when you get T cell activation, the monocytes are not necessarily activated as well. Monocyte and macrophage polarization really influences what happens to macrophage gene expression and virus gene expression. And it may be that in a rather inflammatory response, you have macrophages that are not going to rebound. What it also means is that if rebound and reactivation is going to clear the T cell reservoir, it may not touch the macrophage reservoir. That may be very different from reactivation of the T cell reservoir. So I think what we see here is, while the T cell reservoir and the macrophage reservoir is at the same level, reactivating one and the other are going to be very different. So I just want to, again, say that with the QVOA, we identified functional latent reservoirs in 2 models of ART suppression. CD4 T cells and monocyte macrophages represent, at least in the SIV model, functional latent reservoirs of cells that need to be studied and addressed during the cessation of ART. Rebound studies suggest that rapid ART suppression correlates with slower viral rebound when ART stopped. And in 2 models of SIV ART, suppression and rebound of CD4 T cell reservoirs increase during rebound without a detectable increase in the monocyte or macrophage reservoir. Thank you. Oh, sorry. I forgot the most important thing. All our collaborators, the Silicano lab, everyone in our lab, the entire retrovirus lab, and our students and fellows, our technicians, NIMH, NIAID, and NIDS, and all the companies that graciously really give us free drugs to treat our animals. Thank you.
Thanks so much, Janice. We have time for 2 questions, if anyone has any. The microphone will be coming around. Guido had one.
Hey, Janice, thanks for your great talk. Very, very interesting. I think there's no question that macrophages are involved, in virus replication. And so my question is, how do you think they maintain the virus for such a long period of time? So the reason I'm asking is because when Mirko Baiardini and I did that experiment that you probably saw, published a few years ago, and we put those CD4 depleted animals with a ton of virus in macrophages and almost no CD4s in the system on therapy, they had this rapid decline of virus replication after ART initiation, which suggested, a short in vivo lifespan of the productively infected macrophages. And I was wondering how they maintain their infectious status over time on ART. I mean, are they doing homeostatic proliferation like the CD4 central memory cells?
No, we don't think I think that when they infect in the tissues, so we're looking at the alveolar macrophages, which again are the embryonal derived macrophages. Those cells don't die. I think you do see monocyte-derived macrophages dying and being killed. And there's a lot of those in tissues, but I think the resident macrophages in brain and lung and spleen and gut, probably, the ones that are resident there don't die. They're different macrophages. The other thing is macrophage expression of virus really is very different from T cells. When macrophages are really in their inflammatory state, they downregulate virus. Whereas where they're in the resting state, it's a more permissive state. So I think we have to understand the biology. The biology of the macrophage is completely different than the T cell. And that's why I thought it was surprising that we saw the same level of latency. Whether that's maybe that's the virus and not the cell, right? And so, I can't explain your results. I just know that even in our our dual infected model, at the end of infection, we still see, even though that we lose a lot of cells, we still see that the macrophages are there and infected. So, and that's why we moved to the SIV-251 model, because many people are using that or the 239, and they're pretty comparable. And our model was a standout. Ours was very different. And so we felt we had to do the other model to really validate that what we were seeing in this unusual model was true in the 251 model.
Thank you, Janice. That was a beautiful talk. I was wondering, have you, with your quantitative virus outgrowth assay with macrophages, have you done multiple simulations to see if you can get out additional viruses with a second? And I appreciate maybe the difficulty the macrophages don't live long in culture. But have you tried to do a second or a third round of activation to see if you get more intact proviruses out?
What we have to do with the macrophages is that we actually take supernatants off every 2 to 4 days. and then we actually quantitate them. And what we see is macrophages, unlike T cells, where you get this exponential growth. What you see is you'll get a blip of replication in the macrophage, and then the next supernatant might go down, and then it'll come back again. So I don't know if that's subsequent. We haven't gotten to the point of sort of splitting out the cells, because they're not like T cells. We would have to actually trypsinize them and take them out again.
But what might be interesting is if in a well, and I see what you're saying there, in a well that was negative for outgrowth with a single round of TNF, if you were to add an additional round of TNF, do you get macrophages to spit out?
Well, we actually put fresh TNF every 2 days in.
I see what you're saying.
Because we actually do that, but it would be an experiment to not do that and then see, right? But there's, we've only been doing this for about 2 years, so there's a lot of more that we need to do to characterize it, to understand it, and to, to look at it in depth. You had a question?
Yeah. Unfortunately, I was wondering in one of the last slides you were showing ART released and the lack of change in macrophage reservoirs, but when you look, just without ART release, just longitudinally at the macrophage reservoir, what do you see?
We see it like the T cell reservoir. It's stable. So long term, we've looked at animals now a year and a half on ART, and as I said, we can take spleen biopsies and we do blood, and we see that there's a stable reservoir, but like in the T cells and in the macrophages.
For how many years?
Well, they were a year and a half.
Okay.
Yeah.
Thanks so much, Janice, and thank you everyone for the excellent questions. Our next speaker is Guido Silvestri. Dr. Silvestri is professor and vice chair for research in the Department of Pathology and Laboratory Medicine at Emory University School of Medicine. A physician scientist, Dr. Silvestri further holds the title of Georgia Research Alliance Eminent Scholar in Comparative Pathology and is division chief of microbiology and immunology at the Yerkes National Primate Research Center. Dr. Silvestri has been involved in studies of AIDS pathogenesis, prevention, and therapy since 1993, working mainly with SIV and SIV-infected non-human primate models. Dr. Silvestri directs the Race AIDS Translational Research Program at Emory, which focuses on finding a cure for HIV. Today, he will share work on CD8 T cells and HIV SIV eradication. Please welcome him with me.
Good morning, everyone, and thank you very much for the kind invitation. It's a great pleasure to be here with many friends and good friends in the audience. And so I'm glad that Janice gave this beautiful introduction to NHP work so that I can go straight to some of the data that I would like to share with you. I'm going to talk about some already published data, and I'll try to make that relatively fast. And then I will try to spend more time on unpublished data that I'm sharing with you that hopefully would be of some interest. The whole point of this talk really is to try to figure out what CD8 T cells do in the establishment and the maintenance of the reservoir in the setting of HIV or SIV infection treated with antiretroviral therapy. And there are two premises that I want to make very briefly. One is a specific premise comes from someone who's been involved in evolutionary studies of SIV infection. You probably know that I've been studying for about 20 years the natural infections of African monkeys that have high virus replications but don't get sick. So I've been sort of very, very much thinking along the lines of what evolution of the host-virus interaction has done in the setting of HIV and SIV infections. And one thing that I would like you to keep in mind is that HIV and SIVs never evolved to adapt their host-virus interaction in the setting of antiretroviral therapy. they evolved very nicely to avoid pathogenesis, and they evolved very nicely to escape the immune response. But they never ever evolved until maybe basically a few years ago to deal with the constant suppression of their replication and therefore persisting in vivo for many years without having their replication being allowed. And I think this is an important point in our understanding of cure-related research that not often is sort of spelled out, and I like to spell it out. The second thing is that the complexity of what the CD8 cells do, and this is something that I sound like a broken record, but there's been, we have studied CD8 a lot based on their function as cytotoxic T lymphocytes, their ability to kill, and we know that CD8 T cells do that. They express killing molecules like perforin, granzyme B, and other molecules. They certainly can mediate this effect. It's very, very clear. It's been shown in hundreds of studies, maybe thousands of studies. However, that is not the only function of CD8 T cells. There is a huge wealth of data coming from mouse immunology, from human immunology, suggesting that CD8 T cells do also other things. I'd like to remind the audience that when CD8 T cells were originally described by the group of Schlossman, Reinertz, and others at Harvard in the mid-'80s, they were called suppressor T cells, because the first phenotype that was identified attached to the CD8 cells was to suppress T cell activation in certain experimental conditions. And there is very interesting data on the innate function of the CD8 T cells that has been developed in many mouse models. And I would like to point out, particularly for those who are interested in the work that has been done by Dr. Bedoui at the Doherty Institute in Melbourne using the Salmonella typhimurium model, in which it's very clear that control of this infection in vivo is mediated by innate non-cytolytic functions of the CD8 T cells that are restricted, class 1 restricted at the level of antigen recognition, but completely innate at the level of effector mechanism. So please do keep a mind open on what CD8 T cells do, and please do not equate CD8 T cells with CTL activity. There is a strong Venn diagram, but not all CTL. Not all the CD8 cells do is the CTL. So with those premises, I'd like to go into what CD8 T cells do in the setting of, how do I go forward? Oh, this. Is this one? Oh, should I use this one? Oh, okay. See. That's definitely more efficient. So what CD8 T cells do in the setting of the natural history of HIV and SIV infection is very clear. There are so many lines of evidence suggesting, indicating very, very clearly that the CD8 T cells suppress virus replication. I think as a non-human primate researcher, to me the ultimate demonstration of that is that when you deplete the CD8 T cells, in vivo, you have this bounce back of virus replication that happens pretty much wherever you do it. You do it in acute infection, you do it in a chronic infection, you do it in elite controllers, non-elite controllers. It's very clear. As soon as you remove the CD8 T cells from the system, virus replication bounces up 1 log, 2 log, 3 log. So there's no question that it's one of the few areas, like I like to say, that is not controversial in the field. Something that we described, my student Emily Cartwright described and published a couple of years ago, is that when you treat animals with antiretroviral therapy and then you perform a CD8 depletion, what you see is that virus replication is fully suppressed, virus production is fully suppressed, and once you deplete the CD8 T cells, there is you start seeing bleeping of virus in plasma, and when the CD8 cells come back, this bleeping goes away. And we have done that in numerous animals. There were 13 animals in this paper, and every one of them did the same exact thing. These animals were treated between 8 and 32 weeks, so between 2 and 8 months of antiretroviral therapy. So it was sort of a relatively short to medium time of antiretroviral suppression. But it was interesting that all the animals showed the same pattern. This is, again, published work. I want to go fast. One point of that work that was interesting to us is that when we sequenced, we did SGA and sequenced the virus coming from those blips versus the virus that was present in the inoculum versus the virus that was present at the initiation of antiretroviral therapy, which was day 56, 8 weeks after infection. We noticed an interesting phenomenon that, and there are 3 animals in the paper, we did it in few more animals after that, that the virus that blips after CD8 depletion is more closely associated with the virus that is present in the inoculum than the virus that has evolved in those 56 days in vivo during natural infection, possibly as a result of some kind of immune escape. The other point that I wanted to make is that this is a paper that just came out in Journal of Virology a couple of weeks ago, is that once you deplete the CD8 T cells, you induce an increase in CD4 T cell activation and proliferation. This is something, again, non-controversial, has been shown in many, many studies. As a matter of fact, the first study that Norm Letvin and Jörn Schmitz did 20 years ago, they observed this very phenomenon of the activation of the CD4 coming from the CD8 depletion. So one question is that are the CD8 cells doing something, or is just a passive consequence of having more CD4 cells that are activated? So to answer that question, we did a series of experiments for a total of 8 animals that we just published in Journal of Virology a couple of months ago, and where we induced, deplete the CD4 T cells in vivo in animals that were treated with antiretroviral therapy. And by doing so, after the depletion, the phase of depletion of the CD4, you induce a quite robust proliferation and activation of the CD4 cells, which is clearly homeostatic in nature. And when you do that, the interesting finding is that you have no blipping of the virus whatsoever. So the CD4 activation induced by CD8 depletion is associated with virus reactivation. The CD4 proliferation induced by CD4 depletion in the presence of the CD8 T cells does not lead to any virus blipping whatsoever. So suggesting that it's something that the CD8 cells are doing, not just a passive consequence of the CD4 cells becoming activated. So the explanations for what the CD8 T cells are doing under ART are multiple and may involve some kind of CTL activity. In other words, that there is constant reactivation of some viruses and the CTL go in and kill them, and once the CTLs are not in the system, the virus will be detectable. This virus production will be detectable. Another possibility, which I thought was interesting, is that the CD8 T cells can mediate some kind of transcriptional silencing of the virus. This is a bit of a non-canonical function, non-conventional function of the CD8 T cells, which has been actually proposed many years ago by the group of Jay Levy and his colleagues with some interesting in vitro experiments. But what really sort of fascinated me about the possibility are 2 things. One is the nature of the virus that comes out of the CD8 depletion, the fact that it's old virus rather than the virus that was present at the time of ART initiation. And the second thing that attracted me to that hypothesis is an old study that we did many years ago in tandem with another paper published by Satya Dandekar, in which we tried to measure the half-life of the productively infected cell in vivo in SIV-infected macaques with or without the CD8 T cells in the system. And what both studies found for a total of 17 animals and about 30 measurements is that the productive lifespan of an infected cell appears to be very similar with or without the CD8 T cells in the system, suggesting one possibility, of course, is that CTLs are killing very early on the cells before they produce virus. You can make a lot of scenarios, but clearly one scenario is that some of the components of this suppressive effect are non-cytolytic in nature and don't act on the lifespan of the productively infected cells, but rather act on how many cells are actually producing virus in vivo on any given time. So again, pointing at some kind of silencing of virus transcription. So to test this hypothesis, we decided to do an experiment, because one prediction, if this thing is true, one prediction would be that a latency reversing agent is much more effective when the CD8 T cells are not in the system. If there is this constant tone of suppression of virus reactivation and virus production by the CD8 T cells. So to do that, we decided to focus on an interesting molecule, which is called ART-803. Actually, the new name is N803 because Altor has been bought by NantKwest. I had to change the slides, but please forgive me. So the N803 is a superagonist of IL-15, and IL-15 is a really cool molecule that we and others have been interested in for many years. But in this setting, it's interesting because IL-15 appears to be and has all the credentials to be an effective latency reversing agent for CD4 T cells, and has shown some effect in that regard in vitro. But when it's given in vivo, doesn't seem to have any real LRA activity, and in fact we confirmed that in our monkeys. I'm going to show you in a second. But what I was curious about, okay, what's going to happen if we do IL-15 treatment when the CD8 T cells are not in the system? So we designed this experiment that you see here. It's a reasonably powered experiment with a total of 35 monkeys. So 7 monkeys on top were just given the IL-15 superagonist, and 14 monkeys were given CD8 depletion alone, and of those 14 animals, 7 got a cycle of ART-803 at the end of, during this phase of CD4 reconstitution. And that rationale for ART-803 was sort of a different rationale, was to actually boost the CD8 response at the time of CD8 reconstitution to see if that would do something. And then the third group of animals on the bottom were given ART-803 at the time of CD8 depletion. What I would like you to do is to focus on the left side of the graph and the first phase of the experiment, because in the first phase of the experiment, there were basically only 3 groups, 7 ART-803, 14 CD8 depleted, and 14 CD8 depleted plus ART-803. The right side of the experiment, I would like to ignore it for a second because we can focus on, so that we can focus on the first month or so treatment. So the first thing that I want to show you is that, as expected, when you give ART-803, you get a clear increase in NK and T cell proliferation. This is what others have shown, with IL-15, so there's nothing really surprising. Some of the pioneer work in IL-15 was done by Peter Katsikis and others, Louis Picker, and it's very, very clear that there is this response of the CD4 and CD8 T cells, and particularly the NK cells. One thing that I wanted to point out, I don't have it in the slide, that the CD4 T cell proliferation in response to ART-803 is the same with or without the CD8 T cells in the system. Now, this is exactly that's the result. So I do have that slide, it's right here. And this is just a little bit of RNA-seq data to show that ART-803 was effective in doing the type of IL-15 signaling that you would expect to see in vivo after you give the molecule. So, but this is the interesting stuff. So when you give, so the way you see these graphs on the left is in the axis is y-axis is the SIV RNA copies per ml of plasma. You see that the animals got a very robust peak of virus replication. This is what you get with SIV MAC239. There's nothing special there. We excluded B08 and B17 animals because we are, like Joel, in this study, we're not interested in controllers. We're really trying to look at the natural history. We let the animals go for 56 days, which is different before initiating ART, which is different than many others do. I mean, there's been a lot of people like Dan Baruch, Louis Picker, who have been treating animals after 4, 5, 6 days of infection. We like to do some system that sort of mimics a little bit more what you can do reasonably in humans. So 2 months of unperturbed virus replication. And also, we like to have a system in which the CD8 cells are given some time to mature an immune response against the virus, because I mean, clearly, if you remove all the antigen by day 4 or 5 of infection, you might completely change, the dynamic of the CD8 response. So, in this setting, we treat them with antiretroviral therapy day 56. You see that in the next 12, so we keep them on ART for 12 months, which is a reasonable time. I would like to keep them for 5 years if I could, but the NIH is going to it's not going to let that go, so we'll have to compromise on the length. So what happens is that you see that we use two different virological assays, one sort of workhorse assay with a limit of detection of 60 copies, and then an ultrasensitive assay in collaboration with Jeff Lisson that has a 3 copies detection. So the animals go pretty quickly below the 60 copies. There is a little bit of virus production that remains, and when you go, so the 3 time points before the initiation of N803 where the animals were measured with ultrasensitive assay, and you see that some of them are still detectable with that assay, often in the range of single-digit copies per ml of plasma. What's important, the bottom line of this slide, is that once you give IL-15, an IL-15 superagonist, which is perfectly capable of inducing the IL-15 signaling pathway and is perfectly capable of inducing CD4 T cell activation and proliferation, there is no virus reactivation whatsoever. And this is, I think, quite clear, and has also been shown by others who have used this molecule in ART-treated macaques. This is what we found in the 14 animals that have underwent CD8 depletion. As you can see, when you do CD8 depletion, there is, again, some level of virus reactivation. This is consistent with our previous work. One thing that I wanted to show you, to make you notice, is that in this case, we treated the animals for 12 months versus the 2 to 8 months of the previous experiment. And the level of virus reactivation in general is less impressive. So suggesting that if you keep the animals on ART for a longer period of time, something happens to the reservoir that it's less, the maintenance of this lack of virus production or very low level of virus production is less dependent on the CD8 T cells being in the system. And in fact, some of the animals, you can see the green one, did have a robust virus reactivation, 2 or 3 animals, but others didn't and remain sort of flirting with the threshold of detection of our assay. But this is what we think is very interesting, is that that very Altedo-3 treatment that didn't do anything at all when it was given alone, now given in the setting of CD8 depletion, results in a very robust virus reactivation. And one thing I want to point out is that all those measurements that you see here, all those measurements refer, except for the first one, which is day 3, all the other measurements refer to 7 days after the administration of IL-15. So really, whatever the effect is persistent for 7 days. To give you a sense, this is the relatively famous study by James Whitney using the TLR7 agonist. So if you look at this, you see all those blips, but by day 7, so where the arrows are, by day 7, there's nothing. So if they had done this experiment with the same timeline of measurements that we have done, there would have been nothing shown. So they saw this nice, very, very short, very brief, peak of viral blipping within the first day or 2 after TLR administration, but then everything went back to normal. Instead, in our study, you can see that there is this chronic plateau of virus reactivation. And I have to say that, to my knowledge, this is the highest level of virus reactivation seen in vivo in either humans or non-human primates or humanized mice that I am aware of. And if somebody else knows better, but it's really remarkably robust, remarkably consistent over time among the animals. And the other thing that we're not ready to show the sequence data, but the suggestion from the animals that we've been sequencing so far is that the virus that emerges in these blips is very diverse, suggesting that there is a reactivation of multiple population of virus coming from latently infected cells. So that was the interesting news. The bad news is that when we interrupted those animals, nothing really happened. There were maybe 1 or 2 controllers. You see that green line in the group of Altedo-3 plus CD8 depletion. You see the purple line in one of the animals with CD8 depletion alone. But by and large, there is no control of virus replication after ART is interrupted. And this is consistent with data suggesting that the size of the reservoir in all those interventions is actually not modified. And this is really interesting because when you look back at this graph here, the IL-15 doing nothing by itself, okay? And then you look at this, how much IL-15 does when the CD8 cells are not there. You can think about two things happening here, right? One is that this is all virus that is reactivated and with the CD8 in the system would be killed by CTLs, right? Or you can think about some transcriptional silencing of the virus that is done by the CD8, and now the cells are producing virus that normally would not produce because the CD8 kept them quiet. If this is the case, if the CTL hypothesis is the case, it would mean that in this experiment there is a ton of virus reactivation induced by IL-15, and we don't see the virus because these cells that are reactivating are immediately killed by the CTLs, right? And that's a very sort of rapid dynamic, and we don't see the virus being produced in plasma because the cells are just killed too quickly and too fast. If that's the case, we would have actually achieved the shock and kill that we have been talking about for so many years, and the reservoirs should decline over time. The reason the reservoir doesn't decline, the fact that the reservoir doesn't decline, first can be explained that you have proliferation of the CD4 T cells that compensate somehow for the ones that are killed by CTL. And that's one possibility, but the other possibility, perhaps more parsimonious if you want, and not invoking any coincidence, is that those cells are simply either producing virus or not producing virus, but their number simply stays the same over time. And what we're seeing is just the virus being produced by these cells when they are put in a position of producing virus. So all of this, it's interesting, but one question we wanted to address is this something that can be seen also in the humanized mouse model? And this is part of the collaboration that we're doing with the University of North Carolina, Victor Garcia, and the CARE Collaboratorium. And so Victor kindly, decided, was very intrigued by these results, and decided to do the same experiment in a number of mice, and I believe he has more mice now. But what is striking is that he found exactly what we found. No virus reactivation when Altedo 3 is given alone, some level of virus reactivation where you perform an anti-CD8 treatment, a CD8 depletion, and then more robust and uniform virus reactivation where Altedo 3 is given in the absence of the CD8 T cells. And I have to say it's very nice to see the two models not sort of fighting with each other for what is the best, but actually supporting each other and providing complementary information. So these are some of the future directions. But what I want to show you the last 5 minutes is something in vitro, because what happens is that you do all this work in vivo, and then you start thinking, okay, if there is some transcriptional silencing of the virus, which would be sort of a non-canonical mechanism of CD8 action, can we see that in vitro? So we hooked up with Rafik Sekeli, my former mentor, and Diana Kolpa, who's an assistant professor at Emory, who comes from Rafik's lab as a postdoc. And we used, so Diana and I used this LARA latency and reactivation assay, which is a very interesting assay that Rafik and Diana have developed to look at how latency is established and can be reversed in vitro. And this assay has been presented several times in meetings, but the mainstay of this assay is that you take cells from a negative donor, you infect them with 89.6 HIV, and then you kept the cells in an environment, initially with IL-2 and saquinavir, then with IL-7 and TGF-beta, and antiretrovirals, so that latency is established. And the way this assay works, which is to me very interesting, is that, and I don't want to take any credit because it was developed by others, but the way this assay works is that by day 6 in culture, you still have a subset of CD4 cells that are producing Gag, that are Gag positive. And you can do the same thing with a GFP-expressing virus. And by day 13, those producing cells are pretty much gone, and all the virus is in latently infected cells, right? So what we decided to do is experiments in which we were adding to the CD4 cells Those are memory CD4 cells that are treated with and set up for the LARA assay, different concentration of autologous CD8 T cells. And those are CD8 T cells that have never seen the virus. Those are negative donors, so there shouldn't be any real CTL activity in this system. And what we have done in, this is 8 donors. I believe Diana now has up to 12 donors. Is that what happens is that you have this residual gag production in the CD4 cells, and this is done on memory CD4 cells, also on sorted central memory, transitional, and effector memory CD4 cells. Once you add the CD8 cells, the level of gag-producing cells at the time, day 6, which is the sort of intermediate phase of the LARA assay, is much lower when you add the CD8 cells. And therefore suggesting that the process of establishing latency is accelerated, the curve of establishing latency is shift to the left, is accelerated in the presence of the CD8 T cells. Interestingly, if you look at the number of cells that are actually infected at the end of the assays, looking at the frequency of integrated cells with integrated virus in their DNA is actually higher when the CD8 cells are added. So suggesting that there hasn't been any clearance as expected because those cells are not CTLs. There's been any clearance of the virus. In fact, there is more virus in the cells that are treated with the CD8, co-culture with the CD8, but their establishment of latency has been accelerated. So at least under these experimental conditions, you do have an effect of CD8 T cells, which is related to favoring transcriptional suppression and favoring the establishment of latency. So this is my final slide. I want to spend just 30 seconds if I have them here. I like to try to suggest to think a little bit differently and more broadly about what CD8 T cells can do in vivo. So we have known for a long time that in absence of ART, CD8 T cells suppress, and some of the suppression is mediated by CTL, some of the suppression might be mediated by this HIV silencing. What we know so far, and this is, I didn't want to go into details, but what we know so far is that, so this is very characterized, how the CTL worked. We know that it's class 1 restricted, that is not present in HIV negative subjects, requires cell-to-cell contact, it's mediated by perforin granzyme B, and is certainly subject to escape and exhaustion. What we are starting to learn about this effect, and again, this is work mostly done by Diana Kalpa, I don't want to take too much credit for it, but this activity is class 1 restricted, requires some level of cell-to-cell contact, and appears to be significantly mediated by IL-10 and TGF-beta. So one scenario that comes up is that this activity in the setting of ART actually leads to the stabilization of the reservoir as opposed to the clearance of the reservoir that is mediated by CTL. Now, evolutionary, I started with evolutionary comments. I want to finish with evolutionary comments. You can see that those two activities are actually going in the exact same direction when it comes to the natural history of the infection. They're both aimed at suppressing virus production and virus replication. And in that setting, it doesn't really matter if you do it by killing or not killing, by eliminating the reservoir or not eliminating the reservoir, because there's no art in the evolution, remember, so nothing really matters. What matters is how much virus replication is controlled, okay? Now we have introduced something completely different. We brought ART with our technology in the system, and we created an artificial, if you want, contrived system in which all of a sudden this activity is still good. The CTL activity is still good because it clears the reservoir by killing the infected cells. But the other activity may actually end up being not very good because it could stabilize the reservoir and could inhibit the clearance mediated by latency reversing agents. And I want to finish by just saying that this effect, again, is nothing novel that we discovered. It will be a suppressing effect of CD8 T cells that has been shown in mouse models for many years. And the fact that it's mediated by IL-10 and TGF-beta, it's nothing really new because those cells have been, again, described in many, many other models. With this, I want to finish by thanking a number of people in my division, in my team that have been really supportive, Mirko and Vendi, Steve Bosinger, Cindy, and Paul Johnson, who's our director, but really is a colleague and a friend working with us in these studies. Julia McBrien is the leading student for the ART-treated project. Emily Cartwright was the first author on the first CD8 depletion study. Diana Kalpai-Shaputarin was definitely on this slide, is the person running and setting up the LARA assay. And of course the funding agency, NIH, Emory, CFAR, AMFAR, GRA, and the drug companies to give us literally a ton of drugs for free, which is highly appreciated. Thank you very much for your attention.
Thank you so much for an excellent talk, Dr. Silvestri. I think we're going to have to leave questions for that one during lunch, just so we stay on time. And so we're going to end with a talk on ethics. This is going to be given by Dr. Jeremy Sugarman, who's the Harvey M. Meyerhoff Professor of Bioethics and Medicine at Johns Hopkins. He's also the Deputy Director of the Berman Institute of Bioethics here. Dr. Sugarman is internationally recognized as an expert in bioethics, and in particular, he brings expertise in applying empirical methods and evidence-based standards to this field. In the area of HIV, he's particularly known for his contributions in HIV prevention strategies and, of course, in the ethics of HIV cure studies. So he'll be speaking to us on that topic today. Thank you, Dr. Sugarman.
So while she's trying to get that loaded, thank you for the invitation, and I love that you're intercalating some of the ethics and community bits into what typically can just be a scientific event. And obviously ethics starts with great science. And so it's nice to have them work together and be discussed at the same time, because inescapably when we go into trials involving humans, the issues are going to be very clear, but they start a bit earlier. Part of my interest in this began when I was invited by the IAS to speak at several of the cure symposia that they have before meetings of their annual meetings. But I also, during my sabbatical a few years ago, spent time in Melbourne with Sharon Lewin and her group working on some of these issues. And they also thought it was odd to have the ethics guy down the hall. But it worked out. I actually completed the orientation process much faster because I didn't need to learn all those things to be in a lab, which is good. You don't want me in your lab. All right, so here's the thing. This is the perfect trifecta for ethics. Scientific excitement around the possibility of cure and the related science is really high. And people who are affected by HIV would really like a cure. Treatment is one thing, cure is another. The hope for a cure for many diseases, and especially a stigmatized disease, is really understandable, and it has to be balanced, obviously, by where we are in the stake of the science. And because of that, the moral stakes are really high. So why is there a role for ethics here? For those reasons. We have this great excitement. This is awesome science, and what we're learning about it may or may not lead to a cure as we anticipate it, but we're learning a lot along the way. And we have to do balance the needs of folks involved. So initially, the IAS put together a paper that looked at, Bernie Lowe and Christine Grady, great colleagues of mine, led a working group for the International AIDS Society to look at what the ethical issues might be in HIV cure research. And this doesn't seem much different than any other emerging science, right? And especially emerging science related to HIV. Their first principle was that there ought to be collaborative partnership with the people most affected by the disease. There had to be social value of the research that's moving forward, so not just scientific value, but social value. The work had to be scientifically valid. When you're involving human subjects, there has to be fair selection of participants. The risks and benefits have to be balanced. There has to be independent review. That's your institutionalized review board. Institutional review board. Joke before lunch, come on. There are only so many ethics jokes you can do. I know the food's getting warm back there. I started late, but don't worry, I'll get you. Informed consent, my favorite enzyme, right? So needing informed consent, but meaningful informed consent of participants, know what they're getting into. Respecting enrolled participants and communities. Okay, so this is the starting place. And this, again, like I said, is the part where we start all good science in contemporary day. They also talked about the need to compensate for study-related injuries. That can be quite contentious, especially, as you know, with federally funded research in the United States. All international bodies that look at research say you have to compensate people if they're harmed in research, but we have little clauses around it. But they said, in this high-stakes research, we need to think through the compensation issue. There should be post-trial access to the interventions that are demonstrated to cure HIV, and communities and individuals who participate should receive fair benefits and participation. So this was the sort of starting place where the discussions go, but it gets more complicated than that. The additional issues to think about are the types of research interventions, conflicts of interest, confidentiality, responsible reporting, and the language of cure. And I'm going to go through these in turn. Now, there are many types that we've already heard it today, and there's more to come, about different types of research that's research towards a cure. That's where the language is now moving, research towards a cure. The transplant situation, which drove a lot of this, the proof of principle discussed earlier, Timothy Ray Brown, once known as the Berlin patient. We haven't had the Baltimore patient yet. Boston folks had some, right? We had Mississippi patient, child now, or infant, now baby, now child, now person. We have different names based on geography. That's kind of interesting. I don't know who thought that up. But the transplant idea led to this idea that you could actually have someone who was cured of HIV. And that's a great story, and we're still trying to sort it out. So what are the lingering issues? The assumption at the outset was, if you transplant someone with a mutation in the CD4 receptor, it's going to work. Well, it hasn't worked subsequently, not in an enduring way. And when people rebound, they rebound hard. So the question is, if we are doing different transplant interventions to figure out what's going on, when is it appropriate to do transplantation? So you may remember the story of Timothy Ray Brown. He had a malignancy that required the transplant anyway, which requires ablative therapy. It's not a minor procedure, right? It's getting safer and safer and better, but you don't wake up one day and say, transplant me, please, right? And before we'd do such a dramatic intervention, we'd want to make sure that it was indicated, right? Okay, so we get that part. So what's the bit about towards the cure? What happened there? That's a really important puzzle. So it worked. Was it because he had 2 rounds of ablative therapy? Was it because he had a CSF leak during his second round? Was there a reservoir? What do we learn and what do we know from the times we've tried to tweak different interventions, to modify that protocol in different ways, to say that there's something that we can learn about that was true in the Timothy Brown case, but hasn't been true in subsequent cases? So when a patient who undergoes a transplant with what you think is the right situation, when, if ever, is it appropriate to then withdraw therapy to see if the person's been cured? And that raises huge issues, as Linda intimated at the beginning. What are the issues for that individual subject? What's the rebound like? What's the reseeding like? I'll talk a bit more about ATIs in a minute. But the particular issues there in this patient that's in front of you, Timothy Brown, remember, came off his meds alone, first time, right? Because he just tired of it. Christine presented in one of the best grand rounds ever in the Department of Medicine in Hurd Hall, a patient of hers who decided to stop taking treatment. And patients may decide to do that, but when could we advise or when might we suggest to a patient that that's a reasonable risk for them? We faced that issue actually when I was in Melbourne with a patient who had an opportunity to learn something else that was different. We created a grid of what was known and unknown in his case versus others and came up with ways of offering him that opportunity. Early treatments. Deb's going to talk after lunch, so you got ethics for appetizer and you've got pediatrics for dessert. It's usually the other way around, but so be it. She'll get you afterwards and talk about early treatment and the fascinating questions about the durability of what happens in that case. But neonates obviously can't make decisions for themselves, standard problem in pediatrics, but the risk-benefit calculus and how we think through the ethics of pediatrics is really important. If an adult says, yes, I want you to assess, to do a biopsy, check this reservoir, poke me, prod me, get stuff from me, from every known body part and some that I didn't even know existed, virtual spaces that become spaces. An infant can't do that. Child may not understand that, and what the implications, the safety implications are for a child to advance science and his or her safety is really important. The issues of reservoirs and reactivation, giving people poisons to kick a virus out, that makes people angry when I say that. They're not poisons. Well, in fact, they are poisons. That's why they're kicking these things out, right? You're not treating something, you're trying to kick out a virus. Now, that's really interesting, but it incurs a certain risk with an uncertain benefit. And then the puzzle of the year is the ones on ATIs. So, well, let's just go there. So ATIs are clearly the gold standard. We need ATIs to establish true remission or cure. We have other assays that people are developing. You've heard about them. They fail on certain grounds because the true test is, can you be off antiretroviral agents, and can you have a durable cure? The risks are real to participants and partners without personal benefits, except maybe I'm off this drug for a little while. They're somewhat analogous in research ethics. We always look for analogies to washout studies in psychiatry. So in treatment of long-term psychiatric illnesses, such as schizophrenia, people don't like to be on their medications. They come off their medications, and how do we decide if a new medication is going to work? And you need to do washout, right? You need to remove the drug from their system to see how they do. But the repercussions for people who are requiring that treatment to be stabilized can be often profound and severe. The vulnerability, the inherent vulnerability in psychiatric illness becomes apparent to people when you look at the case. And there have been terrible stories of washouts in psychiatry in which there are suicides, there are violent behaviors and the like that result from the research itself. Now, we don't anticipate that sort of thing in HIV cure and in ATI, but we need to keep in mind that this idea of taking away an agent has been associated in other times with problematic issues in understanding the vulnerability of people who, for whatever reasons, might want to be off of treatment. Well, what are the risks? We've heard some of them. There's some more. There's probably this list could grow. Better bullets. We're in Baltimore, so there can always be more bullets. Viral rebound, acute HIV, opportunistic infections, seeding of reservoirs, resistance, and frustration over failure. And I want to highlight that last one. The idea that you get geared up, you undergo whatever it is that's been proposed. We're all going to watch out for the science sides. I'm not worried. I'm trying to problematize the pieces that you haven't or may not have been thinking about. But the idea of having someone who's been willing to undergo whatever the regimen is, kick and kill, transplant, early treatment, multiple biopsies, assessment of the reservoir, gazillion questionnaires, and they come to the time of ATIs and it fails. It can be hugely frustrating to be that person that also failed science. Failing the investigators they develop deep relationships to, failure to partners, failure of hope. So really an important piece there. The other bit that Linda mentioned briefly is the partner issue and others, partner and partners. The idea of infecting others and changing the dynamics of sexual or drug-using relationships with people. Because the idea is we don't know typically when a rebound occurs, and when that rebound occurs, it comes with a high viral load, and you're pretty transmissible. It's nothing like U equals U. So that bit needs to be really considered about how we're going to think through the dynamics of others who may or may not be engaged in research. There are going to be known others, the great partner who comes to clinic every time for the research setting, and maybe the unknown partners hooked up online or wherever. Those issues are important. For enduring relationships, redefining the relationship that may have been characterized by one of unprotected sex, now being one that needs to be protected in the name of science, warrants some discussion. If it makes you a little uncomfortable hearing about it, it's really important that we talk about it. Agency and understanding. People have to have agency and be willing to take the risk. And understanding their agency and their voluntariness and the permissibility of opting out is really important. So consent begins, voluntary informed consent, in someone who is really positioned to make a voluntary choice. They seek care at places like this. It's fantastic. They get amazing care. But they've developed a relationship, and that issue of being engaged in research is important. Can they say no with the other forces there? You have to ensure ways of ensuring agency. And the issue of understanding is really important. Early on, we were talking about this issue about what we call this stuff and what a cure might mean. And actually the first IAS conference where we talked about this, people were surprised that I mentioned this. The cure was like, couldn't be a 4-letter word, it's a good thing, right? But the idea of being cured for many people means what? I'm cured. I'm not treated, I'm cured. Now the analogy, here again, comes from what? This is October, Delta Airlines, go on it, you'll see more pink than you've ever seen in your life, right? The race for the cure in breast cancer. Okay, so a race for a cure, but there's a popular understanding about the idea that a cure is aspirational in cancer. Cure is a novel thing. It may not be to everybody in this room, or you wouldn't be here, unless you got to the wrong room or you were really hungry, you forgot your lunch, and you heard that there was lunch here. Right? There are different motivations people have. You should say you're interested in cure if you eat the lunch. Just say it, okay? Ethics or others. Okay, but the idea here is what does that mean and how we phrase that, right? Cure initiatives at the NIH, cure initiatives at IAS, research towards a cure is an idea. Now the idea of a remission could be understandable, but could be frustrating. So we don't know precisely what the term is. I don't object to research towards a cure. I don't even object to cure-related research, but in the context of consent and ensuring agency, we need to be crystal clear about what this means. The burdens, typically lots of invasive procedures to help establish safety for and during an ATI. How frequently are you going to monitor people? So in the case in Melbourne we were discussing, the idea was this was in the wake of the Boston patients all rebounding really quickly. And the question was, how frequently would you expect someone to come back for viral load monitoring in a real life situation where it's their personal health and the health of their partner? And the question is, how burdensome is that? I'm sure everyone here enjoys coming to Hopkins each morning and parking. We have a thing you get to slide in the thing and it goes pretty quickly. And we tend to get here early enough that you can get in and find a space. So imagine what it'd be like. How often do you want to come back to where? And what are going to be as new technologies emerge for home testing and the like? It may change, right, point of care testing and the like, what the burdens might be. But how do we best do this to encourage people to get tested as frequently as we think they need without overburdening them and making it impossible for them? In the pediatric setting, especially the idea of invasive procedures to get at different reservoirs, most of those reservoirs are tough to get to. And they require scopes and things that are just not what you wake up in the morning thinking about. And if you do, don't say that at lunch. But volunteer for some of the studies. We can use more healthy controls and not healthy controls and the like. Anyway, the issue here also is selection of participants. Who gets selected? If we expect HIV cure research to work where it's most needed, where would that be? In a place in the world where treatment is widely available? Or would it be where treatments are unavailable? So if you were to think about where the application would be from a public health perspective, where it would be most important would be places where you can't get drug to people, where it's too hot to maintain the stability of a drug. You'd want to be thinking places that are very different than here. People of different races, different colors, acquiring HIV by different means, but fairness needs to be considered so that it's appropriately applied. So implementing them, as ATIs are considered, they should be justified scientifically and ethically. So there should be an explicit scientific justification about what we expect to learn in this situation. It doesn't mean that the intervention is necessarily going to work, but there's got to be value add to contributing to the scientific database. And it should be ethically justified as well. There should be procedures to ensure agency, to ensure that people are capable of consenting, to making sure they're positioned to make a voluntary choice, that they give consent. Okay, and that involves understanding the risks, benefits, alternatives, procedures to be followed, and all those things, but in a way they can understand it. The slide sets from this morning, as good as they are, probably are not going to be a good information exchange mechanism during a consent process. Right? It's hard to get to people and help them understand where this sits in the understanding of the world. Really close monitoring, privacy. The issues that we call people by their cities It's important, right? But what will we do in a high-profile case of the next person who's likely to be a cure? Timothy Brown made the decision for himself to out himself in terms of his name. It was kept the Berlin patient, was an interesting idea. But what can we do? Because you know we're going to have a good press release as soon as you guys crack this case. But what are we going to do in terms of maintaining privacy? And how do we select people fairly? The additional issues, conflicts of interest. This stuff, so conflict of interest, totally separate talk, but I got to tell you, there's a lot of money here, right? And I'm glad we get a lot of it. And the NIH has been giving us a lot of it. That's great. And we want more of it. That's great. And drug companies very interested in this, right? There's a lot of money at stake. So the issue is not that money's not transferring hands. We can't do great science without a lot of good money. That's perfect, but we need to disclose the sources, right? We need to disclose what we're getting, how it's funded, and how decisions are made with respect to our science. Confidentiality I raised as well. Responsible reporting is really key. What I'm worried about here, I'm not worried about when we get the home run. I'm more worried about responsible reporting of results that weren't as anticipated or negative results. In this field that poses real risk to people, the things that don't work, and the field's been pretty good on this. I mean, the Boston description, of the problems that are encountered, great. But we need to continue it because there are probably a lot of other cases of people post-transplants for a variety of settings that aren't reported because it just didn't work. We need that aggregate information. We need to overcome that barrier of the fact that it's only the New England Journal who wants a positive publication. Get over it. It's only a regional medical journal, right? Publish it someplace good, But seriously, get the information out there so that we can understand it and share it. And if no one will publish it some other way. Everybody here is facile enough by publishing information. Say, I submitted this 5 times. It couldn't get published because it was negative. Here it is. Have at it. It may not contribute to my tenure and promotions, but have at it. See, Janice left so I can say that stuff. Okay, and finally, be sensitive to this issue of the language of cure because it may have unintended and intended consequences. So, what I hope I've done is to show you to help meet the moral and scientific promise of HIV cure research. It's essential to address explicitly the related ethical issues, and approaches to managing them warrant careful public deliberation and engagement. Like Linda mentioned, there's a couple of workshops and ideas to try to come up with standards for how best to do this. And finally, when we get to larger scale projects, Christine mentioned in the introduction, some of the things that some of us do in the Berman Institute is to use the tools of the social sciences to help inform ethics deliberation. So what we do is we monitor and assess and engage different stages of the process. And when we ramp up enough, there'll be opportunities to actually measure things and to help make sure that patient-reported outcome measures are there, including ones related to the ethics of the research. So thank you very much for your attention.
Fabulous job keeping us on time for lunch. I think we can take a question or two if anyone has a question for Dr. Sugarman.
What's for lunch? I don't know. Nobody's brave enough.
Or if anybody has a question for Dr. Silvestri, since we picked up a little bit of time, thanks to Jeremy. Okay.
Thanks for this. Yeah, that was a very nice talk, and I was just curious about the right arms of your experimental design. What happens downstream? I imagine that. So after the CD8 depletion, you have very nice reactivation of the virus when you had IL-15 complex. Then, sorry. Oh, sorry, I thought it's not working. No, it's working. Okay. So then, afterwards, if you collect the CD8 T cells, you see the clearance of the virus, or is still ongoing?
Yeah, I wouldn't use the word clearance. I mean, the lack of production detectable in plasma. But yes, when the CD8 cells come back, the effect of Altedo 3 is not there anymore. So if you look at the potential bleeps in the group of, you're referring to the group of animals that underwent CD8 depletion first and then got the Altato-3 later on. So those animals don't bleep, basically. We gave the Altato-3 at the time the CD8 cells are coming back, and there is no, I mean, there's some initial, bleeping, but by the time the CD8 cells are reconstituted, there is no more. They're not, they go back to like the animals that were treated with Altato-3 without being depleted, basically. I also want to take 1 second to apologize for my going over with my talk. I thought, honestly, I thought that was the last talk before lunch. I should have been more careful. Sorry about that.
No apologies necessary. So thank you everyone for a fantastic morning. Hopefully we've planted some seeds, and during lunchtime and the poster session, you can all have some productive discussions. We'll meet back here at 1:15 to get started on time for the afternoon. Thanks.